Okay, in this video we're going to talk about protein synthesis. So protein synthesis all begins with genes. So of the approximately 10,000 genes that make a human, basically they all code for some form of protein. However, these genes are made of exons and introns. Exons are the part of the gene that actually code for amino acids. Introns are the non-coding segments that are interspersed among those exons. So when we talk about post-transcriptional processing, then we're talking about how we have the shuffling of exons from a gene. So RNA is this go-between molecule that links DNA and proteins. Now, RNA copies from the DNA code and nucleus, and then that copy of RNA is carried to the cytoplasm, to ribosomes, where those RNA copy is used as the blueprint for protein synthesis at the level of the ribosome. So all RNA is formed in the nucleus, and RNA differs from DNA in terms of its structure. Remember, RNA has uracil instead of thymine, and RNA has a ribose instead of a deoxyribose sugar. And RNA is typically single-stranded rather than double-stranded like DNA. Now there are three major types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA, or mRNA, rRNA, and tRNA. Now, messenger RNA is this single-stranded RNA molecule. It codes from the DNA template, and it's copied that's complementary to the base pairs of DNA. So what we find is that messenger RNA is a copy of the DNA gene, and this process is referred to transcription, where DNA is copied into the mRNA template. Now, mRNA maintains this triplet code or codon from DNA, and each triplet code encodes for a specific type of amino acid. Now, ribosomal RNA, or rRNA, is a structural component of ribosomes. It's found where protein synthesis occurs, and along with tRNA, it helps to translate messages from messenger RNA into a polypeptide. So messenger RNA brings in the instructions, tRNA has the recipes, rRNA really helps out the whole process. So transfer RNA, the reason why this has the ingredients is that this is the carrier of amino acids. So this has special areas that contain a specific triplet or anticodon that binds with messenger RNA. So it turns out that the anticodon of tRNA is complementary in terms of base pairing with mRNA codons. So anticodons on tRNA match up with codons on mRNA, remember the triplets. And if this occurs at the level of ribosome, so that messenger RNA has the instructions, tRNA's got the ingredients, so these match up, and eventually that makes your growing polypeptide chain. Now this process is referred to translation because you're basically translating the information from, a nucleic acid into polypeptide. That's why it's a translation. Now transcription is where you have DNA that's encoded into mRNA. Translation is where mRNA is decoded into polypeptides. So we see this process here. This is showing the central dogma theory of protein synthesis. And we have the DNA double-stranded within the nucleus. Remember, you can copy a specific segment of gene within DNA to make your pre-mRNA. We have RNA processing where exons can be snipped out and recombined to form messenger RNA. And then messenger RNA can travel through the nuclear pore over to a ribosome where the messenger RNA brings in the instructions for protein synthesis, and then transfer RNA or tRNA brings in the amino acids that link up together to form a growing polypeptide chain. And this is actually going to be specific to this specific mRNA sequence. So transcription is this process of transferring code held in DNA genes to mRNA. We actually have transcription factors or protein complexes that actually activate transcription. And this is done by loosening histones. That way DNA can be read. And you can do this by binding special sequences of genes to be transcribed through this thing called a promoter, which is like the starting point. And this only occurs in the DNA template strand. Now, the thing that binds to the promoter is called RNA polymerase, and RNA polymerase is an enzyme that synthesizes mRNA from DNA, and it starts at the promoting region. Now, what we see then is that once your mRNA or polymerase binds to the promoter, it's going to copy one strand of the DNA molecule into an mRNA template, and then the RNA polymerase will unbind once it reaches this stop codon here. and we have this completed mRNA strand. Now you'd call this pre-mRNA because it's just recently been transcribed. And so it still can undergo post-transcriptional processing where you could potentially cut out specific pieces of this mRNA and recombine them to form a different strand of mRNA. But what's interesting is that these are all exons which came from the same gene. So in terms of processing of mRNA, this is where this newly formed mRNA is edited and processed. We call this pre-mRNA. And remember, introns are removed by spliceosomes, leaving only the exons, and then even exons can be removed or shuffled around to form kind of different mRNA strands. Now, in terms of translation, this is a step of protein synthesis where we have our messenger RNA, which carries the particular genetic code. We have tRNA, which carries the amino acid ingredients, and then ribosomes would be like the kitchen here, where basically it serves as the place where it all comes together. And we have different events that occur during translation. And sometimes this occurs on rough ER, where those ribosomes might be attached to the endoplasmic reticulum. So remember, our genetic code is basically our mRNA strand. Each mRNA strand has lots of codons, which are triplets. And remember, mRNA came from the DNA gene, in your nucleus. Now, there are 64 possible codons because you're talking about combinations of A, U, C, and G. Now, because there's 3 different places in a triplet codon, to take the probability of combinations, all you do is take your 4 different nucleotides to the power of 3. It gives you 64 possible combinations of triplets. Now, there are 3 of these 64 that are stop codons, which means that they code for a stop signal. So during translation, when the protein's being synthesized, once you reach a stop codon, the protein stops being made, and this is what prevents the amino acid chain or polypeptide from being extended any further. Now, there are only 20 possible amino acids, though. So we have 64 combinations of codons. 3 of those are stop. So I mean that leaves 61 other codons. However, there are only 20 different amino acids. So because we have 61 different combinations of codons left, only 20 different amino acids, this means that there are multiple codons that can encode for the same amino acid. Now, this redundancy helps to protect against transcription errors, and it's just a nice way to ensure that proteins are made properly. So remember, tRNA has your amino acid, and this is where it's attached to this strand of RNA. This section of tRNA is called the anticodon. And you have U, A, and U nucleotides here. These anticodons are specific to a codon, and you'd find that the codon is located on the mRNA sequence, so that this tRNA would be complementary to a codon, and it brings in a specific amino acid that correlates with that specific type of codon on mRNA. Now remember, tRNA binds this specific amino acid at one end, and this is now called this aminoacyl tRNA. And the anticodon is basically what binds this triplet code that binds to the codon on mRNA. Now, the anticodon of tRNA will only bind to the codon on mRNA that's complementary. So, for instance, let's say if your codon on mRNA is AUA, then the only tRNA anticodon that could bind with this is UAU. So these would be complementary, where A's match with U's. And remember, we don't have T's here because we're talking about RNA. Now, ribosomes coordinate coupling of mRNA and tRNA. That's why the ribosome is like the kitchen in this process, because it kind of helps put it all together. And the ribosomes contain 1 binding site for messenger RNA and 3 binding sites for tRNA. The aminoacyl site is used for incoming, this aminoacyl tRNA. The peptidyl site for tRNA is where you have linkage to the growing polypeptide chain. And then we have an exit site for outgoing tRNA. So looking at this here, what we find is that we have our mRNA strand that has woven through different ribosomal subunits here. And as this mRNA strand goes through the ribosome, essentially this ribosome is able to read the information along the strand and then grow out this polypeptide chain. Now, as the polypeptide chain is extended, it starts to fold due to its biochemical properties. And remember, the specific sequence of amino acids here in the polypeptide chain determines the specific structure of this protein, which then also determines its specific function. So it's important to note then that this genetic sequence helps, in part, eventually to determine the function of this protein. And what's cool is you can actually see this under electron microscope. So you can see that here are the ribosomes, and then if you look closely, this little strand is your mRNA strand. Now, termination is the third step here, where one of these stop codons, like UGA, UAA, or UAG, enters the first site of the ribosome. And this is what stops translation. Mostly because we actually have specific anticodons here for these codons, but they always code for stop. The protein releases, and essentially what we find is that this causes the release of the polypeptide chain, as well as separation of the ribosomal subunits, and then subsequent degradation of that mRNA. That way you can get rid of the signal, and you don't have a bunch of instructions building up in the cytoplasm. The final polypeptide can be further processed by, the rough ER and even your Golgi body in order to make your final three-dimensional protein structure. This slide actually summarizes the process we just described, where essentially you have your mRNA strand that leaves the nucleus. It eventually finds a ribosome, and it starts to wind through that ribosome. Remember, mRNA strand has triplets of specific nucleotides here, which we call codons. This is a start codon here, which is AUG, and that brings in a methionine here, which is your starting amino acid. Now, that's the initiation step. Now, the next step here is called elongation, where amino acids are added one at a time by other tRNA molecules. So that other transfer RNAs come in, they bind to the A site, and a new amino acid is added to the growing polypeptide chain. And this will continue until we end up having the termination step, where we have a stop codon, this UGA, UAA, or UAG. All of these are stop codons, which is basically a stop signal. This causes a release factor to bind, which allows for your polypeptide chain to unbind from the ribosome. The ribosomal subunits separate, which frees up your mRNA strand, and then this mRNA strand is actually degraded by RNases, or which are enzymes that break down RNA in the nucleus. So the role of rough ER here in protein synthesis is that once that growing polypeptide chain basically dissociates from the ribosome, we actually have a signal recognition particle on endoplasmic reticulum that directs messenger RNA ribosome complex where to dock. Now, once this is docked, this polypeptide enters the endoplasmic reticulum, and that sugar groups are actually added to the protein as it's elongating, and this shape can be altered. Now, protein is then enclosed in a vesicle and transported towards the Golgi apparatus. So what we see here then is we have, with our growing polypeptide chain, at the very end here we have a signal recognition molecule, or SRP, and as this growing polypeptide chain is basically forced into the rough ER cistern, sugar groups can be attached by different enzymes, and basically this modified polypeptide then is packaged into a vesicle, which then can be sent over towards the Golgi apparatus for further processing. Now, in terms of just a summary of DNA to proteins, remember, complementary base pairing directs the transfer of information of DNA all the way to protein. So that DNA triplets code to mRNA codons base pair with tRNA anticodons, and these tRNA anticodons make sure that you have the correct amino acid that's placed on the growing polypeptide chain. And what we're saying here then is that we have complementarity between our polypeptide chain and the DNA that initially coded it.